Hey CW Apes, Mr. Kennedy here. Today we're going to take a look at a lab through a slightly different lens. This lab is called our Toxicity and LD50 Lab. Now, this is normally a lab that could take a good couple of weeks for us to set up, run, record data from it, all that kind of good stuff like that, and then clean it all up. We don't have 2 solid weeks to sit here doing this, so Mr. Kennedy has pulled the documents from our webpage for you, and I've got some sample data for you to use to generate the lab report and still kind of like interpret how all of this works. So this experiment's handout can be found on our class webpage in Unit 5 under Land and Water Use. You want to find the LD50 Lab on our class webpage. And then in your notebook, you're going to generate a lab report that has all this good stuff in it, like what's the question, purpose, background, hypothesis, materials. You're going to make an experimental design, data table, graph, and then as you get to the bottom here, you bet there's some analysis questions, conclusion, error analysis, and the like. So this is a full lab report that you're going to deal with in your science notebook. Now, since you're not completing the experiment yourself, you need to pay very close attention to what you see and hear in this video so that you have all of the tools that you need in order to complete the assignment. The idea in an LD50 experiment is for you to pull off, well, a very simple evaluation of a toxic substance, we'll call it chemical X, to determine the lethal dose for 50% of the individuals in your population. In this case, our population is going to be made up of a bean called a mung bean. Mung beans, well, they're kind of like bean sprouts. Some people eat them, and, well, I hear they're quite tasty. Well, in this case, we're going to look at their sensitivity to a very simple everyday material that you guys eat probably way more than you really even want to or should, and that is salt, table salt. So we're going to perform a cereal dilution. No, not breakfast cereal. Cereal as in a series of dilutions to determine what level of table salt is toxic to 50% of our mung beans. Why do we care? Well, this is a huge idea in the agricultural community, especially if you live anywhere like, coastal or even like the west side of Fresno that is theorized to have once been an ancient ocean and has lots of salt in the soil. Salt can be fatal to plants even in small concentrations. And if you live someplace coastal, well, coastal communities have to struggle significantly with what we call saltwater intrusion. As the community builds up and people pump water out of the ground to water their plants or, I don't know, just take a bath or drink, that creates a suction under your home, bringing the fresh water up to your home and kind of creating a vacuum underground. That vacuum, if there's salt water nearby, like the ocean, will draw seawater in to replace that fresh water. That seawater then comes to the surface and, well, let's be honest, it's not going to be fun for the plant. It's analogous to what you see happening to snails and slugs when they get exposed to salt. The salt in the soil creates a hypertonic environment that literally just sucks the plant dry, and it's very good at killing plants. We want to avoid that. We want to know. We want to be able to test the soil before any plants start to die to protect crops, to protect people and other wildlife from the negative effects of saltwater intrusion. So let's shift gears and look at how we set this up. Then I'll give you some sample data for you to do the number crunching with. So let me move this out of the way and I will show you our cereal dilution PowerPoint. So remember, a cereal dilution is a stepwise dilution series that we use to figure out the concentration at which 50% of our population will perish. So how we set this up, if we were in the lab, we would basically take some little jewelry bags, Ziploc bags. We would cut paper towels to rest inside of them so that they would hold the moisture. Run a line of staples across the bag to hold the seeds in place, And then we'd add, well, 10 to 15 seeds per bag. It's really up to you if you're designing the experiment yourself, how many seeds you want to put in a bag. I usually try to put somewhere, like 10 and call it good. Okay, it's a nice round number and easy to do statistics with, and I, don't even have to take my shoes off because I can count on my fingers 10 seeds, right? That's great. Now, how to do this serial dilution? Well, we're going to start by making a stock solution of salt water. It's a 0.2 gram per mil stock solution. Now, if you haven't had chemistry, or if I just said, hey, go make a 0.2 gram per mil stock solution, some of you might freak out a little bit, but it's actually easier than it sounds. All you need to do is take 2 grams of sodium chloride, that's table salt, and add that to a beaker of 10 mils of distilled water. Easy. Okay. Once you've done that, you've got a stock solution of 0.2 gram per mil table salt. There you go. Now, next step, we're going to get a whole mess of test tubes so we can do our serial dilution. You want to get as many test tubes as you need to match your jewelry bags that you're going to set up. I think probably somewhere in the neighborhood of 8 ought to do it. That's interesting. There's 8 on the bottom of that slide. Go figure. It's almost like I planned it that way. Wow. Don't forget, one of your test tubes has to be your control. So if we're talking about a controlled experiment in this case, I bet that test tube doesn't get any salt water, does it? Okay, so how do we do the dilution? We're going to take 5 mils of plain Jane water and we're going to put that in each of our test tubes. Why? Because it makes doing the serial dilution easier that way, and I don't have to think about, I got to add this many grams of what and where and here and there and all that. That's why. So each test tube gets 5 mils of water. Next, I'm going to take 5 mils from my stock solution and I'm going to add that to the first test tube. So basically I've taken 5 mils of stock solution and I've mixed it with 5 mils of water. So if you're doing the math, if you're paying attention, I've cut the concentration of my stock solution by 1/2. Whoa! That's what I'm going to do test tube after test tube all the way down the line until I reach the end. So once I've added my stock solution to the first test tube, I'm going to mix it. I've now got one test tube that has 10 mils of a mixture of plain water with my stock solution. I'm going to take 5 mils from that first test tube, transfer it to the second test tube, mix, and repeat until I get to the end of the line. Look what happens, right? 5 mils from the first tube, add to the second tube, mix, and repeat. And I'm going to keep doing that until I get all the way to the end of the line. Now, if you've been paying attention, each time I mix and transfer, I'm cutting the concentration of my solution by 1/2. I don't know how much salt's in each of these. I want you to do that calculation. You need to know that this is 0.2 grams per mil. That's my start. And then each of these tubes has taken that and cut it in 1/2. So this one is 1/2 of 0.2 grams per mil. Its neighbor is 1/2 of whatever I had in the first tube. And that one's 1/2 of whatever I had in the second tube. And this one's 1/2 of whatever I had in the third tube. Do the math. I want you to be able to calculate that. Now, as I get this span of concentration in my salt, the idea here is for me to be able to find a spot at which point, well, 1/2 of my seeds die. There will be some points along the way where none of my seeds die, some points along the way where all of my seeds die, and that magic point that I'm looking for where 1/2 my seeds die. That's the LD50. And if I'm an environmental scientist working in the field, I should be able to take a soil sample, like measure the salt concentration, compare it to an LD50 experiment like this, and tell the farmer, tell the people, hey, everything's fine, or whoa, the salt concentration's getting really close to this LD50 level, and we're going to have some problems real soon. That's why we do it. Okay, so we're going to then take this liquid and we're going to add it to all of our bags. All of our like little jewelry bags with our seeds in it. And after that, we get to sit back and watch and let nature take its course. So each bag, each little bundle of seeds is going to get exposed to salt water. And then We just need to count how many seeds grow, right? And then graph that data. Because we don't have 2 weeks to wait, Mr. Kennedy has some sample data for you here that I'm going to share with you. And as we take a look at this sample data, right, and get into that, the thing you need to do is to make sure that you write all these numbers down so that you can do your data table and graph, with these numbers. So everybody's going to have the same numbers. Here we go. Okay, so this is LD50. Here's my bags, the number sprouted, the number not sprouted, and the root length. So if you need to pause the video for a moment to get these numbers down, by all means, go right ahead. But these are the numbers that we're going to use and then calculate our LD50 from that. All right, gang, so that's how to do a serial dilution. That's the experiment. This is your sample data. It's now up to you to generate that lab report and see what conclusions we can make about the LD50 for table salt and its effect on mung beans. I hope that you learn a lot through this. It's a really, really important concept when it relates to saltwater intrusion. I'm Mr. Kennedy, and I will see you next time.